So, hello everybody, I'm Silvia Panarello, predoctoral researcher in the MCS Medicinal Chemistry and Synthesis Group at ICAC CSIC, Barcelona. And I want to say that I'm very thankful to have been selected for explaining my project in an oral communication today. The development of selective photoswitchable pumps for the metabotropic glutamate receptor subtype 1 is a work done under the supervision of the doctors Amadeo Echevarria and Xavier Gomez Santacana. One of the main research topics in which the MCS group is focused is the photopharmacology. Let's see then the principles behind the classical pharmacology and the high precision photopharmacological approach. Pharmacology relies on the systemic administration of an active agent which acts on the part of interest, but also on the other parts of the body. This leads to side effects which results in having a non-selective drug. What happens instead if we are in the presence of what is called a smart pharmaceutical? We have a drug which exists in two forms, the inactive one in the graph depicted in blue and the active one in purple. The radiation with lights of suitable wavelengths allow us to change the drug activity and in most cases in a reversible way. So photopharmacology is characterized by the administration of an inactive drug first and reversible and local activation with non-invasive light later. Now the drug is in its purple active form of acting just in the part of the body of interest. The advantages of this approach are many. Light can be delivered with very high spatiotemporal precision, for that we can reduce the dosage of the drug, very limited effects on the rest of patient's body, and higher efficiency. Moreover, with a final inactivation step of the same compounds that can be photochemical again or thermal, we could limit the problem of environmental toxicity and antibiotic resistance. So usually azobenzene moiety is chosen as a photosensitive element that, when incorporated into the structure of therapeutic agents, should provide control over their activity with light. Azobenzene molecules can be switched generally by using UV irradiation from a flat trans isomer to the bent cis isomer. The latter, being thermodynamically less stable than the former, will isomerize back in time to the initial state. This reverse process can also be achieved using visible light irradiation. Importantly, the two isomers show the measured difference in their shape and polarity, and therefore the light-induced isomerization will result in the switching of the property of an azobenzene modified drug, which may consequently change the drug biological activity. And the spectra here are reported of trans and cis azobenzene, and also they are distinct but overlapping. So azobenzene photochromes have been used in photopharmacological projects due to their small size, efficient photoswitching, large change in geometry, and low rate of photobleaching. Last but not least, for their easy preparation. There are several light strategies used to control protein function. However, the one concerning this project is characterized by reversible properties. The target the target is an endogenous receptors, and the ligands are not covalently attached to the target protein. That means they are freely diffusible. Going deeper, the target of this project are G protein coupled receptors that regulate many physiological processes and are sensitive to a wide variety of endogenous ligands, including neurotransmitters, hormones, amino acids, ions, and several other bioactivity molecules. In particular, we are interested in class C or glutamate family of GPCRs, which represent promising drug targets for a variety of psychiatric and neurodegenerative central nervous systems disorder. The metabotropic glutamate receptor comprise 8 different subtypes, and different studies in recent years suggest an involvement of subtype 1 in learning and memory, but also intellectual disability and autism, And very recently, NGLU1 was regarded as a major schizophrenia and bipolar disorder too. As class C GPCRs, NGLU receptors are dimers, and each protomer includes 3 domains: a transmembrane domain, a cysteine-rich domain, and the Venus flytrap. The Venus flytrap is a large extracellular domain formed by 2 lobes, defined a cleft where the endogenous ligand binds. So this is so-called orthosteric sites, and the binding causes a conformational change of the transmembrane domain that permits the activation of a G protein downstream. So the orthosteric binding sites for a particular endogenous ligand are often highly conserved, making it difficult to achieve high selectivity for specific GPCR subtypes. An alternative and highly successful approach was the development of selective allosteric modulators of the specific receptor subtypes. These small molecules do not bind to the orthosteric ligand binding site, but instead act as at an alternatively located binding site, the allosteric one, which is distinct from the orthosteric one, and where the orthosteric ligands do not bind, to either potentiate or inhibit the activation of the receptor by its natural orthosteric ligand. Then from here, the objective of this project, the development of selective photoswitchable PAMs to reversible control the activity of an endogenous GPCR NGLU1 with light. And why positive allosteric modulators? Because 2 recent independent studies identified 12 rare deleterious mutations in the GRM1 gene, which encodes NGLU1 in schizophrenic patients. Actually, this is the very first study that correlates NGLU1 modification with the schizophrenia. The third work of Ko and Garcia Barrantes demonstrate that these mutations are indeed loss of function mutations that can be restored with novel, highly selective NGLU1 PAMs. And starting from this consideration, we want to achieve the reversible functional restoring with light-regulated positive allosteric modulators. This work has been shared by 2 research centers, the Institute for Advanced Chemistry of Catalonia and the IGF, Institute for Functional Genomics in Montpellier, where the chemical part of the project was developed at ECAP, while the pharmacological one at IGF. The project is divided into several steps. As you can see, it starts with the photoligand design. Then we pass to the multiple step organic synthesis and subsequent photochemical characterization of the compounds of interest before getting tested pharmacologically. At IGF, it was expected to continue with the in vitro characterization, and generally, if the in vitro results are promising, we continue with in vivo characterization. It's not so easy to reach this point. So very often we stop and try to improve the initial design, going back in an iterative way. However, as potential candidates for this kind of application, the molecules have to have precise characteristics like a biocompatible wavelengths of absorption, different isomers population between dark and light condition, and different activity affinity on the target protein between trans and cis. Specifically, these compounds were developed after applying the azologization approach to a known NGLU1 PAM by introducing an azobenzene group instead of N-phenylphthalimide moiety to obtain photoswitchable NGLU1 PAMs in their trans configuration. In order to access an initial library of possible photoswitchable NGLU1 PAMs and survey the SAR for these 3 regions, we decide to maintain the 3-methyl furanylamide as 5-member heterocycle in the molecular scaffold present in the VU series, deputed to enhance NGLU1 PAM potency. So at this point, the photocontrolled analogs were expected to display mGlu1 PAM activity in their trans configuration, and after illumination, the cis isomers should not be active due to the abrupt change of geometrical disposition, and this constitutes a trans-on approach. To offer these 6 analogs plus the non-photoswitchable parent compounds, I use a 5-step synthesis first and later a 2-step one. Next, we determine the photochemical properties of azo compounds by the UV visible absorption spectroscopy in the dark and after illumination with different wavelengths. While in the dark, only the trans isomer is present. Upon illumination with different wavelengths, a mixture of cis and trans isomers can be detected, which is known as a photostationary state. But the illumination with the 380 nanometer light allows us to obtain the highest percentage of cis isomer in the photostationary state. And all the 6 compounds can be back isomerized to their thermodynamically stable configuration by using 455 nanometer light, or allowing them to thermally isomerize in the dark. Moreover, all the compounds displayed high photostability over the application of multiple light cycles. Once all the compounds displayed the same behavior, they were tested for their pharmacological activity as mGlu1. Thus, the dose response curve were generated, and both in dark and 380 nanometer light conditions. And from this set of analogs, the compound 9D, also called MCS 0242, provide the most relevant results with moderate activity as mGlu1 PAM in dark condition. Its potency is clearly lower than the parent compound here depicted in dark. But after illumination, the compound displayed the highest light-induced shift on the PAM activity with a 1 log-fold shift of the potency. Further pharmacological characterization told us that the compounds could display agonism by itself in the dark with a very low concentration of the orthosteric ligand, while upon illumination it does not. Then, in order to understand any undesired off-target activity, we also assessed selectivity of the 6 analogs versus the key anti-targets for this chemotype, the other group 1 mGlu receptor mGlu5 and mGlu4. So as you can see in this graph, in particular, the MCS0242, that is the best kit of this first series, show that is basically unable to activate or inhibit the mGlu5 receptors if compared with the corresponding reference standard compounds, PAM, respectively PAM and NAM. So as mentioned before, these compounds were also tested in mGlu4 subtype, and in particular, the most potent mGlu1 PAM turned out to be an equipotent mGlu4 PAM. It's here. So it displayed, however, a remarkable photo-induced potency shift in both receptors. In summary, while mGlu1 potency could be attractive for a proof of concept for the precise control of mGlu1 receptors with light, the overall profile and selectivity versus mGlu4 were not appropriate for an in vivo translation of possible mGlu1 PAM2 compounds. However, a dual mGlu1 mGlu4 PAM activity could be intriguing for an antipsychotic agent, as the mGlu4 PAM activity can alleviate catalepsies that is a major adverse effect with the standard treatment with antipsychotic drugs. However, achieving these first results led us to further explore the structure-activity relationships around the chemotype of the compounds that we name photoglurax-1 to obtain photoswitchable and selective Anglo-1 PAM activity. Therefore, we synthesized analogs to survey the impact on Anglo-4 activity of different substituents just in the external ring of the structure. And in the only two, we investigate the role of the substituents in 2 positions and also in 3 positions with also adding also 5 chlorine and 4 fluorine always in the external ring. These structural modifications were designed to decrease activity at Anglo-4, trying not to impact negatively on that of Anglo-1. So 9 new photoswitchable analogs were synthesized with the 2-step synthesis. And here again, as you can see, I performed the screening assays of wavelengths to be sure that 380 nanometer was the best to achieve the highest percentage of cis isomers, as well as I repeated the multiple cis trans isomerization cycles. And regarding the photopharmacological characterization, again, these compounds displays an activity over Anglo-1 receptor in the micromolar range. And again, with applying the 380 nanometers light, the light trigger the decrease of activity. Actually, in this case, we obtain a higher light photoinduced potency shift. Regarding the selectivity, the activity towards Anglo-4, we saw that almost all these compounds display the same range of activity as for the Anglo-1 receptors, but with illumination we have a degrees. All these compounds, except one of these second library, that is the NCS 1186, characterized by the double substitution of the fluorine atoms in 2 and 4 position in the external ring. So interesting. These compounds display an activity, very low activity towards Anglo-4, and even less when we apply the light. So we achieved a compounds with a high selectivity Anglo-1 versus Anglo-4. This leads us to the conclusion. And I want to just highlight the most important. So we obtain, after applying the isologization approach, we obtain 2 series of photoswitchable pumps targeting Anglo-1 receptors. And photoglurax-1 displays we can say that is a dual Anglo-1 and Anglo-4 PAM that's under 380 nanometer light. Its potency is significantly reduced. Both of these compounds, photoglurax-1 and photoglurax-2, are the first photoswitchable Anglo-1 PAMs, and photoglurax-2 shows a very consistent on-off photoswitching of Anglo-1 activity and improves selectivity over Anglo-4. So, as future perspective, we can say that the photoswitching we are thinking about to perform also photoswitching with native Anglo-1 in primary cultures. In vitro, performing also in vitro characterization of photoglurax-2 in dysfunctional mutant Anglo-1, and try to go ahead with further in vivo photopharmacological characterization, as well as try to determine the molecular mechanisms in models of schizophrenia in vivo with mutant Anglo-1. So, and now I'd like to thank the 2 lab groups in which I work and all of you for your kind attention. And I will be happy to answer any question you may have.
Okay, thank you, Sylvia. Very, very nice presentations, very nice results. So there are a few questions. We will only have time for a couple, but maybe afterwards you can answer the rest. So there is one from Guillermo that I'm sure you get asked quite often. He's asking, what is the tissue penetrance of the inducing light used for this type of drug activation?
Sorry, so can you repeat because I don't know if I just chat. Okay, now I got the question.
Yeah, so he's asking about the tissue penetrance of the light that you are using. I guess thinking about when you want to use these molecules.
Yeah, in this case, 380 nanometers is let's say for the tissue is not so penetrant as the right one, as the red wine, but it depends on the target that we want to analyze, that we want to assay. So there are different kind of device. If I still share the screen,
we can still see slides. Yes.
Okay, I can show you. Okay. So some example of devices for phototherapies. Let's say this one that is more used now, but because it's past all the the clinical test, is the photodynamic therapy that is used the red light to kill cancer cells. So it's like more superficial. In the moment we want to go deeper in the tissue, we have to use devices in case that's going more inside the tissues.
Okay, thank you. So there's another one from Javier Garcia. It says, have you done experiments with the azo compounds to prove that these ligands are actually allosteric? For example, radiolabeling experiments with an orthosteric ligand?
Yeah, we not me by myself, but another now postdoc girl in the same lab. She did an extensive work about the allosteric alloswitch 1, that is the first allosteric compounds found for Anglo5 receptor, and they she studies basically especially the rate of dissociation of these compounds. And they are quite sure that these compounds binds to the allosteric pocket, because also the rate in which with which these compounds go out the binding pocket is let's say that is confirmed the allosteric behavior of these compounds, and especially the transform, because these compounds was a trans active compounds, was it had a rate different from the cis compounds, that it was the dissociation rates very high. So okay. This is confirmed the transon approach.
Okay, thank you, Sylvia.
You're welcome.
But there are a few more questions in the chat, so that would be great if you could answer some of the questions during the next.
Okay.
Or so. Okay.
Thank you.
Thank you again for very nice work. Very nice
presentation.
Thank you. So our next speaker will be Adrián Gironda Martínez, who is doing an industrial PhD at Philochem in Switzerland. And he, Sylvia, I think you'll have to.
Yeah, I'm trying to find 9 here. How can stop the sharing?
You stop sharing in the. There should be a red button where you can click.
Oh, if not, I'll just leave the meeting and I go back in a couple of minutes. So okay. It should work.
Okay, thank you, Sylvia. Okay, so Adrián, his talk is entitled "The Power of Big Numbers: DNA encoded libraries as a powerful technology in drug discovery. Adrian, the screen is yours.
Thank you very much. I will try to share the screen. So, okay, can you see it?
Yeah, we can see it. Let's see if you can put it in presentation mode.
Presentation mode.
Great, that's perfect.
Okay, good. So thank you very much for the presentation, and thank you very much for attending to my talk. My name is Adrian Girondo Martinez, and I'm a PhD student at Philochem again, enrolled at the University of Siena. And today I'm going to give you some of the results of my PhD project with a presentation entitled "The Power of Big Numbers: DNA Encoded Libraries as a Powerful Technology in Drug Discovery." The presentation is going to be divided in several points. So first, I would like to give you an introduction of the Phylogeny group and the DNA encoded libraries and how we use this technology at Philochem. And then afterwards, I will move on with the results of my PhD project. So Philogen is a Swiss-Italian company which has a clinical trial management and GMP production site located in Siena, Italy, and which has a daughter company located in Zurich, Switzerland, serving as a discovery center. Philogen has been recently listed on the Italian stock exchange markets, and we have 2 fully owned products in phase 3 clinical trials and current collaborations with many important pharmaceutical companies and academic groups all around the world. So at Philochem again, we based our research activities in 2 main technologies. So the discovery of antibodies using antibody phage display and the discovery of small molecules using DNA encoded libraries. So I can understand that many of you may not be familiar with the concept of DNA encoded chemical libraries. So DNA encoded chemical libraries are collections of organic molecules that are synthesized using combinatorial chemistry and which are individually attached to a distinctive oligonucleotide barcode, which serves as an amplification barcode and readable barcode. To understand the technology, we have to come back to 1992 when Richard Lerner and Sidney Brenner proposed for the first time this approach at the Scripps Research Institute. So they proposed the synthesis of small molecules by combinatorial chemistry onto bits and encoding those small molecules by the addition of short oligonucleotides also into the bits. It was not until 2004 when 2 groups in parallel. So first of all, the group of Professor Daroneri at ETH Zurich and later at Philochem again, which proposed and published the method DNA recorded synthesis, in which you can both have the small molecules display on a single or double strand DNA, or you can have 2 different moieties displayed on 2 different DNA strands. At the same time, Professor David Liu at Harvard University describes the DNA depleted synthesis strategy, which relies on the chemical linkage between both small molecules directed by the self-assembly of the DNA. So the general strategy for the construction of DNA encoded chemical libraries for DELs relies on split and pool combinatorial chemistry. So as an example, I will give you this, I will go through this scheme. So first of all, we have an amino modified universal DNA headpiece that can be reacted with many different bifunctional or trifunctional building blocks. After the reaction of these building blocks with the amino modified DNA, we can encode this building block by the addition and ligation of a short DNA fragment to the nascent DNA. After we encode all the different building blocks with different DNA tags, we can HPLC purify them individually and pull them together in a single fraction. Afterwards, this fraction can be split again in many different fractions, for example, 3, and this cycle can be repeated iteratively. So first of all, chemical modification of the small molecule, and then addition of the DNA tag to encode these chemical modifications. So if we repeat those processes iteratively to n cycles, we are able to construct libraries of up to billions of compounds. So the main advantages of the DNA encoded libraries technology respect to, for example, conventional technologies like high-throughput screening is that actually DNA encoded libraries can be synthesized in a very small scale. We can speak about picomoles to few micromoles of total compounds. And for that reason, they can be stored in a single Eppendorf tube. While high-throughput screening platforms, as you may know, they all need big facilities in order to be able to store efficiently and to screen all the compounds. Moreover, one of the main advantages of the DCL technology is that every single molecule can be interrogated again, at the same time against the target protein of interest, because every single molecule is encoded by a distinctive DNA tag, while in high throughput screening platforms, we have to basically screen every molecule at a time. So in this slide, you can see a typical workflow for the DNA encoded library screening campaign. So we start our campaigns with library designs and constructions. So it's important to notice that libraries can be designed in order to target many different proteins, or they can be actually designed in a focused way, let's say to target a certain type of proteins or a particular protein. After designing and constructing the libraries, we screen them against the protein target of interest using either solid supports or in solution. After several washing steps, the non-binding members can be washed away, and the binding members can be eluted out of the protein. And as I mentioned before, every single compound can be tracked down the information, because we can amplify the DNA barcode attached to it by PCR amplification, and then we can sequence it by high throughput DNA sequencing. After that, we always have this type of results, this plot of results, in which we are able to identify different moieties that are enriched in every single screen campaign. And after computational and statistical analysis, we're able to identify what are the most promising hits. So afterwards, we go for off-DNA resynthesis. So we resynthesize all the possible hits without the DNA tag, and then we perform hit validation activities using different binding and functional assays. So at Philochem, we use the two main different approaches for the construction of DNA encoded libraries. First, the single pharmacophore libraries, which are the standard DEL formats. And those libraries display lead-like molecules, and they have like very big diversity. While on the other hand, we also use a proprietary platform called Encoded Self-Assembly Chemical Libraries, or ESAC libraries, which are libraries in which we display distinctive molecules in both strands of the DNA. So we can say that we display fragment-like compounds, and those libraries are of extreme purity, since, for example, for the simplest of the designs, every single DNA conjugate can be purified individually and then annealed. So now I'm going to move on with some of the results of my PhD project. So the first part of my PhD was basically based on the idea of merging both of the most important approaches for the construction of DEL. So as I mentioned before, ESAC format, which is a library of extreme purity and fragment light, but it lacks a little bit of diversity because of there is no actually combinatorial assembly between the small molecules in the typical combinatorial fashion. While single pharmacophore libraries, for example, are highly pure, are lead-like, and they have bigger diversity, since we are not, let's say, limited by only the combinatorial assembly of those two building blocks. Merging these two formats, we are able to produce what we call the new format ESAC 2+1. So ESAC 2+1 is a high pure library again, because as we mentioned before, those compounds can be individually purified by HPLC. Also, the first step on the strand A can also be individually purified by HPLC. And then to couple the second building block, we always use reactions that were already pre-validated, having more than 75 conversion by LCMS. So those libraries have very much enhanced diversity, and they allow us to perform affinity maturation libraries using, for example, pre-identified hits on the sublibrary P. So this technology was patented, and Philochem is the only proprietary of this technology to be used. So as an example of an ESAC 2+1 construction, we start our DNA encoded libraries building up the sublibrary A. As I explained before, we basically couple the first building block and purify every single conjugate, and then we couple the second building block and we purify the pools to be able to get, in this case, for example, an almost 670,000 members, sorry. And sublibrary B, we couple, for example, in this case, 256 different members to the distinct oligonucleotides. And after self-assembly and filling by Krenov polymerization, we are able to get our new SEQT2+1 library, which is composed by more than 170 million members. So before proceeding with any screening campaign against targets of interest, we always perform a high-throughput DNA sequencing of what we call our naive library. This is performed in order to be able to check the homogeneous distribution of every single molecule in the final library pool. After that, and considering that we always include aromatic sulfonamides as building block controls in our library, and using carbonic anhydrase 9 or CA9 enzyme, we are able to basically validate our design and the construction of our library, performing a screening campaign against this protein and identifying the different sulfonamides that can bind to it. So the direct evolution of the SEQT2+1 technology would be the linkage between both DNA strands. And to do so, we can have many different approaches. In this slide, I just show you 4 of them. So for example, we could link both strands by amide bond formation, but in this case, we can have, for example, possible byproducts due to the coupling reagents. We can also perform, for example, disulfide bond formation, but in general, the availability of defunctional thiols is it's really low. Also, we could linkage both strands using reductive amination, but again, the availability of acid aldehydes and the stability of the aldehydes can be a potential issue for this reaction. And finally, for example, in this here, you can see that we also can perform a copper catalyzed alkylase high cyclidation. But the potential issue of this reaction is that the availability of organic acids as commercial building blocks is not really high. So for that reasons, it's clear that we need new DNA compatible reactions. So during my PhD studies, I focused my attention not in developing a new reaction that can basically go over all those problems, but I wanted to focus on a typical transformation on organic synthesis, on classical organic synthesis, which is the diazotroph transfer reaction. So acids are important functional groups in organic synthesis because we can have different products using them as starting materials. So we can have triazoles like click chemistry. We can have amines using Schmidt reaction, for example, or we can have isocyanates using the Curtis rearrangement. Moreover, the amino acids and the amines are among the most common building blocks for the construction of DNA encoded libraries, and so the functionalization of this amine is highly desired. I have to say that this reaction was not yet described in the context of DCLs. It was described in the context of oligonucleotide chemistry, but not with the requirements of construction of a DCL library. So we perform an extensive reaction development campaign in which we basically use as a model substrate just a typical glycine. And we basically change the buffer systems, temperature, reaction time, equivalence of the diazotroph transfer reaction and the reagent, in this case, this one that you can see on the slide, and also the use of copper as a catalyst. As you can see, after an extensive optimization campaign, we reach our best conditions using borate buffer at pH 9.4, 37 degrees for 1 hour, and using only 20 equivalents of the diazotroph transfer reagent and without the need of using a copper catalyst. So afterwards, we perform an amine scope. And as you can see on these examples, the scope of the reaction is really high and the conversion yields are high to excellent conversion yields, except for the typical non-activated anilines, for which we had less than 5% of conversion. So with these results and with this optimized condition in hand, we proceeded to basically perform the diazotroph transfer reaction and the click chemistry reaction to link both DNA strands of SEQT2+1 in what we call SEQT plus libraries. So SEQT plus libraries are, again, highly pure libraries because we can basically purify again every single conjugate individually, and after the diazotroph transfer reaction, we can also purify by HPLC the acids from the amine starting materials. These new SEQT plus libraries display macrocycle-like structures, and this can help us to target more difficult proteins or to drug, for example, protein-protein interactions. Moreover, these also libraries are with enhanced diversity as well. These libraries can be up to billions of compounds again, and this technology was patented in 2019 at the beginning of my PhD studies. And again, Philochem is the only proprietary of it. So again, as an example of a construction of an ESSAC Plus library, we start building up the sublibrary A. In this case, we have 1,480 different amines that can be converted by a diazotransfer reaction in their corresponding asides, and 69 different acid alkynes, acid terminal alkynes that will link to this DNA strand. After self-assembly and filling, and after click chemistry reaction, we are able to obtain our final ESSAC Plus library, which is composed in this case by almost 10 million members. So as I mentioned before any screening campaign, we sequenced our naive library in order to check the homogeneous distribution of the compounds in the library. And then again, using the typical sulfonamides that are known to bind CA9, we are able to basically confirm the design and the construction of our library by screening campaign. So as a conclusion of this presentation, I would like to highlight that during my PhD studies, during the first years of my PhD studies, I was able to produce a new ESSAC 2+1 library in a dual pharmacophore setting. These libraries can be up to billions of compounds, and they have superior purity. They can be in the rule of 5 or even beyond rule of 5 in terms of Lipinski rules. And they serve us also to perform lead expansion campaigns using pre-identified heats and affinity maturation libraries. Throughout the use of in-house developed DNA compatible reactions, like the diazotransfer reactions, we were able to evolve the ESSAC 2+1 format in what we call ESSAC Plus. And as I mentioned before, these libraries are again up to billions of compounds of high purity and display cyclic molecules that can help us to extend binding surfaces, to target extend binding surfaces, and to target, for example, difficult protein-protein interactions. So finally, I would like to thank very much Professor Dario Neri for giving me the opportunity to perform my PhD studies at Philochem, surrounded by many excellent scientists. I would also like to thank Drs. Florence Seman and Etienne Donquel for their continued support and the guidance throughout this journey, and all the rest of the members of the chemistry team and the small molecule therapeutics team, which are led by Dr. Samuele Gazzamali, and also the former members of the group. And I would like also to give special thanks and to highlight the good quality of this symposium, and to thank the organizing committee for choosing my abstract for another presentation today, and also to all the audience for the kind attention. So please feel free to ask any questions you have now, or you can always contact me on my email addresses, either the company one or my personal one, or you can leave the questions in the chat. Thank you very much.
Thank you, Adrian. Very nice work. Very different from the other presentations we've seen. Yeah. I was wondering, what is the success rate of in the heat validation process? When you have you seen cases where you find the heat, but then when you test the molecule without the oligonucleotide, it doesn't bind anymore? Or
yeah, absolutely. So what we normally do when we perform screening campaigns is that the most common way of using it is using solid supports. So we normally try to screen also the library against only the solid support in order to be able to discriminate, for example, possible interactions with it. We also perform some screenings against typical targets, like, for example, albumin or alpha-glycoprotein that are known to bind different kinds of molecules, that are prone to bind different types of molecules. Doing so, I would say that the success rate is pretty high. Nevertheless, there are always promiscuous structures, like, for example, in my case, during my screening campaigns against another protein target of interest, I was able to identify some hydroxyquinolines, and then actually they were later not really active molecules, since they are like really prone to react with many different parts of the proteins and are not really interesting. So it's important to notice that the success rate can be high if the libraries are well constructed, and if the screening campaigns are well performed in order to avoid these promiscuous binders. But of course, it's not as easy as performing, for example, high throughput screening, in which you individually test every single compound.
Well, the success rate is never 100% anyway. But
no, exactly. I mean, you have different companies around the world claiming more than 80%, 90% success rate. There are other companies claiming like around 50 to 60%. So there is always a discrepancy between different approaches.
Yeah, there's a couple of questions in the chat. Actually, the first one is somehow related to this one. So what are the chances of DNA interfering with drug target binding using this technique?
So normally we use this amino modified DNA, including a long linker, normally it's a PEG linker or a C10 linker, in order to be able to efficiently separate the small molecule physically from the DNA strand. In this way, normally, most of the time, we can avoid the interaction of the DNA fragment with the target protein. Nevertheless, for example, there are differences on screening libraries which are built up in single-strand DNA or in double-strand DNA, because, for example, in single-strand DNA, the bases are more prone to interact with surfaces of the protein, while in double-stranded DNA, the bases are kind of protected one to each other, and only, for example, the phosphate groups can interact. So there's for sure cases in which we have interactions between the DNA and the protein, and that's why in our group first before we move with off DNA synthesis, we always perform also on DNA synthesis and validation. So we have a setting to be able to perform heat validation still with molecules attached to the DNA, so individually attached to them, and in this case, we can also try different DNA fragments, different distances, so we can see what's the influence of the DNA.
Okay, thank you. And one last question, and then we'll move because we're running out of time. How stable is the diazo transfer reagent? Is it easy to work with? So
this diazo transfer reagent was originally published in 2007, if I remember well, as you saw it on the slide, as a tetrafluoroborate salt. Before, it was described as a hydrochloride salt, and this hydrochloride salt was potentially explosive, and also it was not easy to handle. The synthesis was not easy. You couldn't evaporate the solvent till dryness, for example, because the compound can explode. But this compound it's really easy to handle. This tetrafluoroborate salt is really easy to handle, and it's really stable. So you can store it at 4 degrees, even at room temperatures, during like long times. You can also store stock solutions in the freezer for long times, and you don't see degradation of the compound, and you still see quite a huge conversion in the reactions.
Okay, great. Thank you again. A very nice presentation.
Thank you very much to you.
And if there are any more questions, they can still ask in the chat. So we'll move to the last speaker before the lunch break, who is Ana Gomez, who is a PhD student in the Paula Gomes lab in Porto, Portugal. And she will be talking about peptide-based therapeutics to tackle skin and soft tissue infections, antimicrobial, anti-biofilm, and collagenesis boosting activities. Anna, the screen is yours.
Hi, good morning, everyone. Let me try to share. Are you listening to me?
Yes, we can hear you, yes.
Okay.
Okay, perfect.
It's okay? Okay, let's start. First of all, I would like to thank the organizing and the scientific committee for the opportunity to share part of my PhD work that is devoted to the development of peptide-based therapeutics to tackle the skin infections. The skin infections commonly occur during the healing process. The opportunistic bacteria can colonize the open wound and form the biofilms, which are difficult to eliminate and therefore delay the healing process. In the most complicated cases involving the deeper soft tissues, like the complicated skin and soft tissue infections, like infected burns, diabetic foot ulcers, or surgical site infections, commonly necessary the inpatient hospital admission and acute treatment. The current standard of care of these skin infections involves incision, drainage, surgical debridement, and antibiotic therapy. And during the hospital stay, these skin infections could be exacerbated by the hospital-acquired pathogens belonging to the escape group of pathogens that are multi-drug resistant bacteria for which efficient antibiotics are being exhausted,
We all know that antimicrobial resistance is disseminated worldwide, and that represents a major drawback in this line of treatment. Therefore, new antibiotics and new options are urgently needed for the treatment of these skin infections. However, an efficient treatment should also promote a fast tissue regeneration, since it is inefficient in elderly people affected with diabetes or venous chronic insufficiency. So, having that in mind, our group designed a chimera peptide for the treatment of these infections, combining in the same structure an antimicrobial peptide and a collagen boosting peptide. And why? Because an antimicrobial peptide, or orally reported with broad spectrum activity and low propensity to induce bacterial resistance, and a collagen boosting peptide for a fast tissue regeneration through collagen boosting effect. As an antimicrobial peptide, we choose peptide 3.1, a de novo designed synthetic peptide, orally reported with broad spectrum activity, and as a CBP, we choose pentapeptide 4, whose palmitoylated form is widely used in cosmeceutical industry under the name the matrix seal as an anti-aging. So with this construct, we aim to provide antimicrobial protection to the open wound and a fast tissue regeneration through collagenase inducing effect. So we synthesized all the peptides through automated solid peptide phase synthesis, and we synthesized the reference peptides, the antimicrobial one and the collagen boosting peptide, and also their palmitoylated forms and terminal palmitoylated forms. We synthesized the chimeric peptides in both possible orientations and also their palmitoylated forms. And finally, a chimeric peptide with a beta-alanine spacer between the two building blocks. Sorry. We assessed the antibacterial activity of the peptide against 2 gram negative and 2 gram positive bacteria belonging to the escape group of pathogens, and we also assessed the toxicity against human fibroblasts. And as expected, the collagen boosting peptides were inactive against all bacterial strains, while the antimicrobial peptide present a broad spectrum activity as described. The N-terminal palmitylation of the antimicrobial peptide leads to an increase in toxicity and loss of antibacterial activity. Regarding the chimeric peptides, they are also active as the reference peptide 3.1, being slightly more active against gram-negative bacteria. The palmitoylated form of the chimeric peptides also leads to an increase in toxicity and decrease in antibacterial activity. The introduction of the beta-alanine spacer in the chimeric peptide didn't significantly affect the antibacterial activity. Since both peptides are highly active against the bacteria, we move further and we use this chimeric peptide 3.1 pp4, since it is less cytotoxic to the human fibroblasts compared with peptide pp4 3.1, and since it is slightly more, it is selective against gram-negative bacteria. So we tested against, gram-negative clinical isolates, multi-drug resistant ones, against Pseudomonas, E. coli, and Klebsiella pneumonia. And as you can see, the mix were really low, between 1 and 4 micromolar, more or less. And the minimum bactericidal concentrations were found to match the MIC values in all the cases, indicating the bactericidal effect of these peptides against multi-drug resistant isolates. So since the bacteria are present in the wound in the biofilms form, we assessed the antibiofilm activity of the peptides, and we made it in 2 different ways: the inhibition of biofilm formation and the effect of peptides on preformed biofilms. In inhibition of biofilm formation, we used clinical isolates from Pseudomonas aeruginosa, since it is a great biofilm former, and a Klebsiella pneumonia, since it's also a biofilm former better than the E. coli. So the results are expressed in biofilm biomass formed in the absence and in the presence of the peptide at different concentrations. And as you can see, for Pseudomonas isolates, there is no significant difference, indicating that peptide is not able to inhibit the biofilm formation of Klebsiella for Pseudomonas aeruginosa isolates. biofilms, while against Klebsiella pneumoniae, there is a difference and is the effect of the peptide in their biofilms. Regarding the results on performed biofilms, the biofilms of Pseudomonas aeruginosa and Klebsiella pneumoniae were allowed to grow for 24 hours, then were treated it with 20 times the MIC and then allowed it to grow for another 24 hours. And the optical density in the beginning and at the end of the plantonic phase was measured. And as you can see, there is a significant difference in the reduction of biofilm proliferation being the effect of the peptide either in the Klebsiella pneumoniae biofilms. We also assessed the reduction of metabolic activity of the biofilms and reinforced the previous results, indicating that the highest impact of the peptide is against Klebsiella pneumoniae isolates. To get more information about the biofilms and influence of the peptides in biofilms, we performed a confocal laser scanning microscopy against one species of Pseudomonas aeruginosa and against two isolates from Klebsiella pneumoniae. And as you can see, there is no significant difference in the control and treated biofilms with Pseudomonas aeruginosa, while in the Klebsiella pneumoniae isolates, there is a high impact, with highest impact against this Klebsiella pneumoniae isolate through biofilm disaggregation. So at that point, we know that the peptide presents antibacterial and antibiofilm activity. So we go further and we assess the collagenase-inducing effect of the peptide. And we made it in vitro using human fibroblasts. And as a reference, we use the palmitoylated form of the collagen-boosting peptide used in cosmeceutical industry. And the results are expressed in amount of collagen produced per viable cells. And as you can see, for all concentrations, there is no significant differences between both peptides, indicating that with this modification, we are able to retain the collagenase-inducing effect of the collagen-boosting peptide while turning it into a potent antibacterial peptide against gram-negative isolates. So this chimeric peptide is disclosed as a promising lead for development of a topical application treatment for skin infections. And at that point, we want to understand what happens to the antibacterial peptide if we coupling an ionic liquid to its terminus, and what happens to their antimicrobial, antibiofilm activity, and what happens to the peptide stability. So taking advantage of our best chimera peptide, the peptide 3.1 pp4, we attached it to it through click chemistry, the Coop catalyzer alkene-azide cycloaddition. We attach it to it an imidazole ionic liquid. And are you thinking, why an imidazole ionic liquid and why through click chemistry? So the imidazole ionic liquids were early reported with broad spectrum activity against gram-positive and gram-negative bacteria, and also as an antibiofilm agent that could enhance the activity of our chimeric peptide. And because imidazoles were reported with as a dermal permeation enhancers, turning them really appealing for the topical application as we want. And why through click chemistry? The click chemistry is broadly used to introduce triazole rings in the peptides to substitute the disulfide bonds, to attach to the peptides different building blocks or different functional groups. And because it's a chemoselective reaction in mild conditions for a peptide synthesis. So we start the synthesis of the peptide in the solid support. At that time, it was manually. We couple all the amino acids. Half of the resin was cleaved, and we obtained the chimeric peptide as a control. And the other half of the resin were coupled to an azidoacetic acid, obtaining here an acid N-terminus acid peptide. Moving forward to the on-resin synthesis on click with previously synthesized propagyl imidazole, bearing here an alkene moiety, allowing us to perform the click chemistry and obtain the triazole ring connecting the imidazole ionic liquid and the peptide. So from now on, I will refer to this peptide as a chimeric peptide and this one as a modified peptide to get it more easier. So both peptides were tested against reference and multi-drug resistant clinical isolates previously tested for the chimeric peptide. And as you can see here, there is no significant differences in the antibacterial activity, with a few exceptions, but there is no significant differences, indicating that this new modification and this new construct is able to retain the antibacterial activity of the parent peptide, the chimeric one. And since this peptide, the chimeric one, presents a great activity in biofilm formation, we assessed also the modified one, and we assessed the activity in inhibition of biofilm formation and also the effect of the modified peptides on preformed biofilms. And at that time, we only made it against Klebsiella pneumonia multi-drug resistant clinical isolate, for which the chimeric peptide presents the greatest performance. And as you can see, in the inhibition of biofilm formation, there is no significant difference between both peptides, with a slightly improvement here for the modified peptide at the half of the MIC. And regarding the effect on the preformed biofilms, there is a differences between the chimeric and the modified one, but also it remains more or less the same. And at that point, we are asking what happens to the peptide stability, since we make some changes in the structure and actually at terminals of the peptide. And we decided to assess the stability of the peptide against tyrosinase-mediated modifications. And why tyrosinase? Because it plays an important role in melanin biosynthesis in the skin, and because their enzymatic activity has a preference for hydrophobic amino acids. And as you can see here, the modified peptide remains with their mounting during the incubation, while the chimeric peptide is completely degraded after 6 hours. So we were able to say that with these modifications, we can achieve some peptide stability against the tyrosinases. So to end this presentation, the big conclusion is that this work with peptide-based ionic liquids is a promising start to the development of new formulations for a topical application in the treatment of skin infections. So I want to end and acknowledge to my supervisors, to my lab colleagues. They are all fantastic and amazing. And I also want to thank for the collaborations with Lucinda for antimicrobial assays, and for Eva with collagen and cytotoxicity assays. Without them, it's not possible, because it's a multidisciplinary work. And I want also to thank for funding and all of you for your attention. Thank you.
Okay. Thank you, Anna. Very nice presentation. We wait for people to ask questions in the chat. I was wondering, I may have missed it, but what is the target of these peptides? Like, what is their mechanism of action? How are they having their antimicrobial effect? How are they killing the bacteria, basically?
Yeah, we haven't explored it yet, the mechanism of action, but we believe that it's similar for the antimicrobial peptides, since there are peptides. So we believe that is the membrane interactions with the bacteria, but we haven't explored it yet.
Okay, and I guess related to this, have you looked at whether bacteria develop any resistance to these peptides over time? Like if you treat them, or how likely do you think it is that they will develop resistance? I guess without knowing the mechanism, it's hard to predict, right? But... Yeah.
But since they are a peptide, the peptides were described to have low propensity to induce the bacterial resistance. So we believe that they would not be, they don't have that ability, but we don't know. And we don't know also the mechanism of action. We believe that is membrane interaction, but we don't know.
Okay. And yeah, there's a question in the chat, but it was the same one, I think. I was also wondering in terms of the next steps. So if you wanted to develop these peptides further to be a treatment for patients, what would be, is there anything you need to improve, or what would be the next models that you would be using?
We need to also make some assays regarding the stability of peptides, because we only assess against the tyrosinases, but there is a lot of other proteases in the skin, the collagenases, the elastases ones, and we need to check the stability of the peptides in that against their enzymes. And we are performing some studies on the activity of peptides in simulated wound fluids. And what happens to the activity of the peptides in the simulated wound fluids. So there is a lot of work to do. We have a lot of other batch of compounds, similars, that we are working on it. But we want that it could be a great formulation for a topical application in this case of the most complicated skin infections. But I think that we are just starting this process.
Yes, you have to test them. So one last question before we finish this session, and this is also from Guillermo, who was asking first about how these peptides work, and now he's asking, is the activity cytostatic or cytotoxic?
Well, we have already checked the toxicity of the peptides for the first ones, the chimeric ones, and the modified ones with ionic liquids. And at that point, we know that they are toxic, but they are selective. So the IC50, it's not atomic concentration. So we believe that they are selective, but we need to check and assess the in the other cells. We only assessed in ACAT cell lines, in keratinocytes.
Okay, still a lot to investigate, which is great. Okay,
yeah.
So thank you, Anna, again, and thank you to the three speakers of this session. The quality of the presentations was really high, so I think it's going to be really hard to choose the prizes. And so just as a reminder that now we have the lunch break, and we will restart with the workshop at 2:15 in this same session. So the same Zoom session where we are, this is where the workshop is going to take place. So yeah, see you later. I hope you all enjoy your lunch.
And thank you, Matteo. I have to present you. Matteo Borghini is a postdoc student in Department of Chemistry at the University of Pittsburgh in United States. So I would like to thank you quite a lot to be here in this almost European symposium. And he's going to talk about the stereoselectivity synthesis of delta. Is delta, no? Yes. Okay, the delta fluorinated isoleucines exploiting CH bond activation. Thank you so much.
Yeah. Thank you a lot for having me here and for the kind introduction. I'm going to present our latest achievement in the CH bond activation field in our research group. So fluorinated alpha amino acids are very important building blocks, and they find application in a lot of fields. One of them is the drug discovery. Here I reported just two examples of drugs developed by Merck and Bristol Myers Squibb, respectively. And in their scaffold, you can see the fluorinated alpha amino acid, because they can be very useful to improve potency, to reduce the metabolism, to improve the permeability, to get a better toxicity profile. But in other words, they can be very useful to improve both pharmacodynamics as well as pharmacokinetics. They also find application in functional imaging. Here we have some fluorine 18 labeled alpha amino acid synthesized in 60 minutes, thanks to the photoredox catalysis. And they have been used to visualize tumor masses thanks to the positron emission tomography. And this is possible because the tumor cells have higher amino acid uptake. Peptide engineering, here we have buforin 2, which is a peptide with antimicrobic activities. And by replacing the two leucines with the hexafluoroleucine, it's possible to improve the stability towards protease as well as the bioavailability. And this can, of course, apply to protein engineering. But in this case, the introduction of a fluorinated alpha amino acid into the protein can be very useful to study its structure as well as its biophysics, thanks to the fluorine NMR studies. And even though the cryo-EM technology is a great technology, is having a great impact on the field, fluorine NMR studies are, I would say, complementary to both cryo-EM and also the X-ray crystallography. So this is how our project was born. We were interested into the cyclophilin A, which is a peptidyl proline cis-trans isomerase, very important for protein folding. And it is known that cyclophilin A is involved in many diseases. But unfortunately, we don't have a lot of information about that, especially about its behavior during pathological conditions. So we thought to incorporate different delta fluorinated L-isoleucines into the cyclophilin A structure in order to perform some different fluorine NMR studies to determine both in vivo in vitro system as well as in vivo system to determine, in fact, the what's going on. So, and since our research group is involved in, is interested into the development of CH bond activation, we saw the opportunity to introduce this fluorinated side chain at this point, at this position, as well as the methyl group at the beta position with the CH functionalization. And all of these starting from the L-alanine. So in the literature, you won't find many approaches to synthesize fluorinated isoleucines. There are some non-stereoselective approaches, but of course you will obtain all of the four stereomers. There are a couple of asymmetric syntheses, but sometimes the lack of, they don't have a great overall yield. Sometimes they use harsh conditions, and also only the delta trifluoro D isoleucine is reported. I mean, its synthesis. The other one is the kinetic resolution. In this case, this enzyme is able to selectively hydrolyze the amide bond to obtain the desired diastereomer. Of course, the yield is low, and but if you're interested into the into others fluorinated isoleucines, you will probably need to use another enzyme because they're very substrate dependent, and so it sometimes can be very challenging because they are also sometimes they are very expensive. And there are no reported syntheses of the monofluoro, the delta monofluoro, as well as the delta difluoro L-isoleucines in the literature. So we got inspired by the work of Professor Shi and Professor Chen on the CH bond activation. They used this advanced intermediate with the aminoquinoline moiety in order to coordinate the palladium species and kick off the pallada cycle. This directive group is important to activate the beta position shown here. They reported to being able to introduce the different alkyl chain disposition using alkyl halides and usually iodides as well as bromides. And the silver carbonate is supposed to promote the SNN2 reaction at this step, and to enhance the reductive elimination, and also to get rid of this iodide in order to regenerate the palladium catalyst. The dibenzyl phosphate is used in a catalytic amount, and it acts as a ligand for the palladium catalyst. It also increase the solubility of the silver salt, and it promotes the protonolysis step. So our first approach in the synthesis of this key intermediate shown here started with the protected L-isoleucine, which was coupled with the aminoquinoline using this condition. And then the CBZ group was deprotected, and then the amine group is reprotected as a phthalimide. But unfortunately, we weren't able to scale this approach because it has some drawbacks in the deprotection step and as well as in the final purification. So we switched to this second approach, starting from the L-alanine, and which was phthalimide protected, and then coupled with aminoquinoline using this different condition. And we were able to obtain multigrams of this K intermediate. So at this point, we start studying the first CH bond activation to obtain the monofluoro derivative. Our approach was to screen the more common CH bond activation conditions present in the literature, find the one that works best in our case, and then optimize it to improve the yield of the product and to avoid the formation of a dealkylated byproduct, which is very challenging to get rid of it. So in this case, the condition that we selected is shown here, and we optimize it, and then we scale this reaction up to 1.5 gram in order to obtain 74% of the desired product. And then we did the same with the to obtain the difluoro derivative, but as you can see here, the reactivity dropped. The last entry, the last condition, the condition at the bottom is the same that we used for the previous for the synthesis of the monofluorinated derivative, and as you can see here, we didn't get any product. So we needed to use harsher condition, and we optimized that. We scaled again the reaction up to 1.5 grams, and we got 60% of the desired product. When we did that with to obtain the trifluoro derivative, we weren't able to get it. We screened different catalysts as well as different solvents, ligands, silver salts, but we never observed the product. We also try to use more activated substrates as well as different dielectric groups, because when we push this reaction, we got 5% of the desired product and 3% of this byproduct, which is the result probably of a second interaction with a palladium species. So we didn't figure it out yet why we got this result, why the reaction with the trifluoro ethyl iodide doesn't work. It's probably due to electronical effects, but we are still investigating, still working on that. So with the monofluoro and the difluoro derivatives in our hands, we moved on with the second CH bond activation in order to introduce the methyl group at the beta position. This reaction is stereoselective because it has a sterically favored transconfiguration, as you can see here. And since the reductive elimination takes place with the retention of configuration, we ended up obtaining the anti-product. At this point, we had to remove the directive group, and we used very mild conditions to do that. So we first bulk protected the secondary amide, and followed by oxidative cleavage. And we got the carboxylic acid, which was further protected to obtain the delta monofluoro as well as the delta difluoro L-isoleucines in 17% overall yield and we were able to get between 50 and 100 milligrams of them. So in conclusion, we optimized the CH bond activation steps, starting from this key intermediate. With the CH bond functionalization, we were able to introduce the fluorinated chains, side chain at this position. And with the second CH bond functionalization, we introduced the methyl group in a stereoselective fashion. We synthesized both the delta monofluoro and the delta difluoro L-isoleucines, and this is the first time that their synthesis has been reported in the literature. And they will be incorporated into the cyclophilin A structure to perform some fluorine NMR studies in order to understand its behavior during pathological conditions. And I would like to thank the Department of Defense for the fundings, and Professor Peter Wibb for letting me join his research group, and also my coworkers. And that's all for me, and I'm happy to take any question.
Thank you, Matteo, for your nice presentation. This was really nice, clear, and well, explained so well your work there in the United States. And because on the chat there is no question at all, I would like to ask you, do you have some specific work to do later on with these L-isoleucines, like some macrocycles or future work with peptide mimetics in the future?
Mm-hmm. I mean, fluorinated alpha amino acid, in this case leucines, they also have different properties. We started this project because our collaborators were interested in the cyclophilin A, but definitely they can be found a lot of applications in the drug discovery, also because for instance, the difluoro methyl group has very interesting properties. It's a bioisoster of thiols as well as OH and NH2 groups. And it also is a lipophilic hydrogen donor group. So you can use them in plenty of ways. So also probably in peptidomimetics as well.
Mm-hmm. We have another question from Professor Cristina Nevado. She's asking, can you comment on the lack of the reactivity of the CF3CH2 urine? Maybe go back to mechanism slide to see.
Yeah. It's hard to say. As I mentioned, we still don't know why we got this result.
Can you go back to the mechanism slide?
Sorry, what, here?
Yeah. So I'm just interested in how you propose that the perfluoroalkyl group engages with the palladium, because maybe that gives you an idea why certain groups work and certain groups don't work.
Yeah, it's hard to say. We don't know yet. I mean, probably there is an interaction between the fluorine group and the palladium, but it's hard to say. We are currently running different experiments as well as different computational studies, but we still don't have an answer for that. And as you can see, though, for instance, the CH bond functionalization with the monofluoroethyl iodide works very well, but with the difluoro, with just one atom of fluorine more, the reactivity dropped. And with the trifluoro, you weren't able almost to get the desired product. So something happened. The starting material is very stable with that condition, so it's not we didn't observe decomposition. So it's probably something happened between the fluorinated group and the palladium species, but we don't know yet why.
Okay, thank you, Mateo. Because we are a little bit short of time, the rest of the question, if we can, you can discuss it in the chat. I will appreciate. Okay. Thank you.
Okay, thank you.
Good luck in the United States.
Thank you.